Hi, everybody. Thank you, Bruce, for such a generous welcome. And I'm impressed how much you remembered from our walk. I thought you were paying attention to the streets we were taking. Turns out you were paying attention to all my chit-chat. I want to start out with a big thank you. As you get older, I'm going to give away my age, you learn to start your days and the important things in life with what you're grateful for. I want to say thank you for you guys to be in here when it's such a gorgeous day outside. That's already, I think, a huge start. I appreciate your making the choice to be here. For me, it's a little easier because this is where I live. I took this picture. This is what it's like most of the year. It's a nice, warm, sunny day, so being inside one day is not the end of it's not a huge deal, but I appreciate you being there. And I really appreciate Mary Beth's invitation to be with you today, and Bruce Cowan's vision in helping us support research through the foundation. He had the foresight to appreciate the importance of research, and from Tri-State, a huge thank you to you guys as the Tri-State chapter for supporting an additional grant. Normally, the National Foundation only has money to fund 7 new established investigator research grants, but thanks to your generosity, we funded an additional grant, as Bruce mentioned, this year, funded an exceptional investigator, Dr. Liu, who works right here, who's actually working on fat-derived stem cells that could be a therapy for scleroderma. So definitely worthwhile supporting her research efforts, and we're all very excited to see what her progress is. So what I want to talk about is why research is important. You know, we are in a very critical time in that there may be more cuts to funding of research into NIH, which would be very, very detrimental to scleroderma research. Already, scleroderma has very little funding at NIH. We certainly don't get as much money for research out of NIH as other rare diseases. For example, multiple sclerosis, which is not more prevalent than scleroderma, but definitely gets a lot more funding. And so if we were to be subjected to additional cuts, it would be very detrimental to the research community in scleroderma. We are interested in continuing the research until we are put out of business for finding the cause and the cure. But we are also interested in making sure that there's a pipeline of young researchers, physician scientists, investigators, who will continue this for us. And we need them to overlap with us because we need to be able to mentor them so that they don't have a huge learning curve. We don't want to lose the momentum. And so research is very important. So I'm going to tell you about some examples of where research that was done in the bench ended up being important to scleroderma patients, but also give you some information that will empower you when you talk to your congressman or representative as to why research on scleroderma is important. Because the first thing you're going to hear back, well, but it's rare. How many people does it affect? So I'm going to teach you what the right answer is. So research is done in the lab. This is sort of a cartoon of what a bench in the lab looks like, what the research looks like. And to you, it probably looks like this, you know, a bunch of words, a bunch of letters, a soup. But you're going to, hopefully, by the end of this talk, learn a lot more that you can explain on your own to your community, to your families, to your friends. So why is research important? Research is important. I'm going to see if, here we go. I can show you with a pointer. Because the research that we do on a daily basis in the universities, in the academic institutions, at all levels, both clinical, bench research, all of it, is what leads to the discoveries. And those discoveries eventually are picked up by the pharmaceutical companies who then go on to develop the therapies. So without us starting the research, there is no therapies because most of the large companies have done away with the research labs and they rely on us to come up with the initial discoveries. So that's why it's important for the research to continue. I also see research as a puzzle. So as we put the pieces of the puzzle together, we can get a better picture as to what's happening in scleroderma, what is triggering scleroderma in some people and not others, and what could potentially be the right cure for scleroderma. So all of us worldwide who do research on scleroderma contribute pieces to that puzzle, and eventually we piece it all together to solve it. All right. So you all know what scleroderma is. You're very familiar with it, but what I want to point out is I'm going to focus mostly today on this, the fibrosis part, and explain what that is and sort of what we're trying to do about it. And I will talk about some of the other aspects, especially a little bit the immune response in scleroderma. But you will see nomenclature that I hope many of you are familiar with, but if you're not, we're just going to go over it very briefly. So there are different types of scleroderma. There's the systemic form, and then there's the localized form that you might have heard, such as morphia, or when you see linear scleroderma in kids that we call en coup de sabre or other forms. But if we talk about the systemic forms of scleroderma, within that group, there's different variants. So there's what we call the diffuse cutaneous systemic form, the kind that we call the limited cutaneous form, and then when scleroderma happens in overlap with other diseases such as lupus or rheumatoid arthritis or myositis. And what do these mean? So if I were to just explain them very simply, and I'm going to go over that, going beyond what scleroderma means, which you all know means hard skin. Thank you. So this is in text what those forms are, and this is if you look at the picture. So we have what we call the diffuse systemic form, where the skin involvement goes beyond the knees and it goes beyond the elbows, basically, and can affect the trunk, the upper arms, the upper legs. The limited systemic form, it's still systemic, limited just refers to the skin area. So this is below the knees. You can see the shaded areas below the elbows. And then there's the form that we call systemic scleroderma sine, systemic sclerosis sine scleroderma, and that means that there's no external skin involvement, but there's still internal involvement. So often when I ask patients, do you have the limited or the diffuse form, they say, well, I have the systemic. But within the systemic, there's different forms. And the reason I'm saying that, because you're going to see what it means in terms of antibodies. So what is fibrosis, what we're going to focus on? So fibrosis is excess formation of connective tissue. A lot of that means, too much collagen, too much proteins like collagen that have different names like fibronectin and others. Fibrosis can happen in various settings, and sometimes it resolves. In scleroderma patients, it happens, but it really doesn't resolve, and that's what we're trying to understand, why it doesn't resolve. And often you'll see it called as uncontrolled wound healing, because in wound healing, there is a response that involves fibrosis, but in scleroderma, it almost seems that that response just keeps on going. It doesn't stop. And too much collagen means what? Too much collagen means that in the skin, where there's collagen fibers that kind of link up together and cross-link, you can have low collagen, and that's what happens with aging. That's what happens when we start getting wrinkles, right? It's reduced collagen, makes the skin a little thinner, and then less cross-linked. The collagen molecules are less linked together, and that gives the wrinkles. But in scleroderma, it's actually the opposite. It's too much collagen. And there's other circumstances when there's too much collagen. So you can have too much collagen that looks like this these days, but you can also have too much collagen where the collagen is in the skin, and it causes increased thickness in the skin. And then there's the too much collagen where the collagen is in the lungs, and it makes the lungs tight and tougher to expand as you try and breathe in, and it affects the function of the lungs. So that's fibrosis, and that's what too much collagen will do. Why is fibrosis important? So the reason research on scleroderma is important is because fibrosis is not unique to scleroderma. But I call scleroderma the prototype disease. Fibrosis in scleroderma affects the skin, affects various internal organs, whereas in other diseases, the fibrosis affects a single organ. And a study from NIH has shown that fibrosis in general, as a feature, is responsible for almost half of all deaths in the developed world. So now that makes scleroderma even more important from the perspective of research, right? And why is that? That's because fibrosis affects all these different organs you see listed. And you see the examples of other diseases that have fibrosis. In a heart attack, there's scarring in the heart, there's fibrosis. In diabetics, there's fibrosis of the kidneys. In the case of liver cirrhosis, that's fibrosis, whether due to hepatitis, alcohol, anything else. So fibrosis can happen in nearly any organ and is a hallmark of many, many other diseases. What's unique about scleroderma is the fibrosis affects multiple organs, not a single organ like these other diseases. But if we solve the fibrosis in scleroderma, it is very likely that we will solve it for all these other diseases. So here are examples of those diseases. Fibrosis in the eyes. You see it with glaucoma. You see it with macular edema and diabetes and retinopathy. You see it in the lungs with a variety of diseases, including asthma. Asthma is fibrosis around the airways. You see it in the kidney with a variety of diseases, in the heart, in the liver, in the skin. So not unique to scleroderma, but certainly important for scleroderma and the other diseases. All right, so I want to start with examples of where research has been useful for the patients, because these are the kind of examples that will also help you explain to others why is research important. It's not something that just we do on the bench in the lab and it stays there and it gets published in papers. It actually ultimately comes to the clinic and applies to your care. So one of the things I want to talk about is the antibodies in scleroderma. So how many of you know you've had an ANA test, anti-nuclear antibody? Okay. How many of you know if it's positive or negative? Okay. How many of you know what the antibody really is beyond just ANA? 1, 2, maybe 3. All right, so it's important to know that information. If you see outstanding rheumatologists like Dr. Gordon, Dr. Spira, others in the field, they will always test for your antibodies because they know how important it is. So years ago, 9 different antibodies were discovered in the lab. And most of the time, they're mutually exclusive in the sense that each patient has 1 antibody. And what's interesting is in spite of the fact that we only have 9 that pretty much cover all the patients, no 2 patients are alike. So when you know 1 scleroderma patient, you know 1 scleroderma patient because everybody is unique. All right, so we test these antibodies by getting a blood sample. So we get a blood sample that has your antibodies in it. So I can see, you can see it here. We put cells on the slide. These are human cells. We add your serum, which has these antibodies, and let them attach to the cells. And then we use something that has a fluorescent tag that will glow to figure out where they're binding. And that's how we do the ANA test. So this is your typical ANA test. And what it looks like is something like this. This is a positive ANA. So you can see these are cells, just hundreds and hundreds of cells on a slide. They're glowing green because that's the tag we used. You can see that it's mostly the nucleus of the cell that's really bright, that's really staining green. And we can tell what the pattern is. And from the pattern, we can predict what the antibody is. So what are the main antibodies that were initially described? So this list shows you the main antibodies right here. SCL70, RNA polymerase, U3-RNP, U1, PMSCL, THTO, and centromere. The most common out of these 3 are anti-centromere, anti-RNA polymerase, anti-topoisomerase. And what you can see is they occur across the world in different populations. So it's not just in the US, it's not just in Europe, it's not just in Caucasians or African Americans, but it's also in other populations, although the proportion of each antibody in each population is different. So for example, we see in Caucasians that these are the 3 major ones, as I mentioned, the ones in red. When you look in African Americans, the 2 that are more common are these. But when we go to the Asians, to the Japanese, you can see that there are 2 that are most common. So it changes from one population to the next. But they still associate with the same features. So the importance of discovering those antibodies is they allow us to predict what kind of problems with internal organs patients who have them might run into. So again, this is not where you say, I have this antibody and I'm definitely getting this problem. Not at all. We just look at association. So maybe 10% of patients with this antibody might get this complication, but it helps for us to estimate or predict or be on the lookout for symptoms of these complications. So ACE inhibitors came up years ago, and ACE inhibitors are now used to really avoid having major kidney problems in scleroderma patients. Kidney failure used to be the major complication in patients and a major cause of death years ago, but no longer is because now there's drugs for it. So the major problem that has come up as a result of kidney being better managed now is lung involvement. So whether it's lung fibrosis or pulmonary hypertension, that's currently what we're focusing on because it is more common and more serious. So when we look at causes of death in patients, and again, you don't take that home as, you don't walk out of here saying, I'm gonna die. This is supposed to be how to empower you with information so you are on the lookout for things and you understand how the research is helping you. So pulmonary hypertension, pulmonary fibrosis are the major problems right now that we don't have effective treatments for. We don't have cures for, and that's what we're working on. Pulmonary fibrosis starts out with inflammation and leads to this fibrosis or scarring in the lung, and usually happens more often in patients with the diffuse skin involvement, although not always. It's usually more severe in African Americans, and it's more common in patients who have this antibody, the SCL70, when we talk about the list of antibodies. We're gonna go, so if you look at patients who have no pulmonary fibrosis compared to those who have pulmonary fibrosis, those who have pulmonary fibrosis do a little worse than those who don't have pulmonary fibrosis. Makes sense, right? You have an internal organ complication, you're not gonna do as well as someone without any internal organ complication. But now if you divide the patients based on those antibodies, so if I separate those with this antibody versus this one versus this one, a pattern comes out as to who does better and who does worse. So THTO is this antibody where the lung fibrosis is worse in patients. So by knowing your antibody, your rheumatologist can estimate which internal organ involvement you may get. Not necessarily will get, but may, but they can also predict how bad it might be and be able to be on the lookout for early symptoms so that you can manage it early. So this is how scleroderma happens. When you look at patients who have the diffuse form of scleroderma, so those of you who have the more diffuse, you know your skin fibrosis, your skin thickening happened very quickly early on. But then you get to a point where it plateaus and becomes kind of steady. And in some patients, it even starts reversing or softening after years. If you have the more limited skin involvement, the skin that's affected is more limited, and the increase is very, very, very gradual. And if you're going to have internal organ involvement, these are the kind of types that might happen, not in everybody, of course. And this is around where in the path it may happen. So we know that in patients with the diffuse form, if they're going to have really bad internal organ complications, it's going to happen fairly early in the first few years, and not as much later. But what we also know is SCL70 antibody, also known as TOPO1 or RNA polymerase, are more likely to happen with this form, whereas anti-centromere and THTO are more in the limited form. So again, trying to sort these unique patients into subsets that help us predict what's going to happen to them. So this is what it looks like. These are the antibodies we're talking about. These are the different ones. And what you can see is that they associate with certain things. So patients who have the anti-RNA polymerase, we now know from the studies that were done in academic institutions, are more likely to have kidney issues. Those who have centromere, a small percentage of them, but nonetheless, some of them are likely to have pulmonary hypertension. Again, this is not everybody. Those who have SCL70 or what we call TOPO antibody are more likely to have interstitial lung disease. And so by studying large populations of patients, we can determine who is likely to get what. But again, it's not 100% of people. It's a percentage within that group that may be at higher risk for developing these complications. So it's important to know your antibody to be able to anticipate what may or may not be symptoms that are important to look at with your doctor. So as I mentioned, lung fibrosis is now an important cause of death and health issues in patients with scleroderma. And that's why we sort of focused a lot of our research efforts now on it. And lung fibrosis comes in different patterns. So when a pathologist does a biopsy of the lung and looks at it under the microscope, there are a lot of different things they can call it, and this is sort of what these things look like. But the most common patterns in scleroderma are what's called UIP, which is usual interstitial pneumonia, or NSIP, which is nonspecific interstitial pneumonia. NSIP is the most common in scleroderma patients. And if we compare scleroderma patients here who have pulmonary fibrosis or lung fibrosis compared to patients who have idiopathic pulmonary fibrosis, so another disease where the fibrosis is restricted to the lungs, we see patterns come out that differentiate the two. So we know for scleroderma, it's more common in women, but for IPF, the similar illness basically that only affects the lung, we know it's more men who are affected. We know in scleroderma the age is younger, whereas in IPF the age is older. We know the prevalence is somewhat similar, and there are other features that differentiate the two, but IPF is definitely a more severe disease because survival is much lower. So how does that help us for the research? So we look at survival and how many people are affected. But most importantly, we'll look at what can we do to discover differences between the two diseases and similarities that may help us solve the puzzle for both and come out with therapies that will be helpful for both. So this is actually a scleroderma lung, and I did not kill anybody for it. This is actually a patient who had a lung transplant. And often when they do the lung transplants, they allow us, if they give consent, the patients will allow us to use the old lungs that are removed, that are fibrotic, that are diseased, to use for research. And that's actually very helpful. So we got a scleroderma lung, and we decided we're going to look at what can you see in common between scleroderma and this other disease, IPF, and can we learn something from this other disease that will teach us something about scleroderma. So what we did is we took the lungs of IPF patients who also had a lung transplant. We took the lungs from scleroderma patients. We took the lungs from patients with a condition called idiopathic pulmonary arterial hypertension. So that's another condition where patients get the PAH similar to scleroderma patients, but it's a unique disease where the PAH is the only feature. There's no skin fibrosis, there's no other features. But again, we could learn something from it that will teach us about PAH and scleroderma. And we took a bunch of lungs that were from donor lungs that were not used for transplant surgery, from normal donors basically whose lungs did not get used. And what we did is we looked at all the genes that are known, thousands and thousands of genes, to see what was turned on in scleroderma compared to these other diseases, and what was turned off in scleroderma compared to these other diseases. And the data looks something like that, and I don't expect you to know that, because if you already know that, I can recruit you to work in the lab right away. But basically, it shows me the color green and red show which genes are turned on, which genes are turned off. And we identified a long list of genes that are either turned on or turned off in all these different diseases. And this allowed us to summarize it this way. So we now know that there are genes that are turned on or off in the same direction in all scleroderma lung tissues, whether patients had pulmonary fibrosis or pulmonary hypertension. So those are genes that are in common in those two manifestations. And if we can target them, then maybe we can solve both complications. We know there was overlap between genes that were turned on and off in pulmonary hypertension in scleroderma, but also in this other similar illness called idiopathic pulmonary hypertension. And we know that there are genes that were turned on or off in scleroderma with pulmonary fibrosis that were also the same as the ones in this other condition, idiopathic pulmonary fibrosis. But we also noted that there are genes that are unique to each of these diseases. So the ones that are not overlapping, where the circles don't overlap, are unique to each. So that helped us come up with a list of genes that basically told us what are the general systems or networks that are affected in scleroderma and other lung fibrosis versus unique to scleroderma. And those are the genes that we can now look at to see, okay, what can it teach us that would be helpful in a clinical setting for the patient? We also found genes that were similar in the lungs and the skin. And that's important because you want to try and develop a therapy that will both improve the skin and the lungs and other organs. So you don't want to end up on one therapy for the skin, one for the lung, one for the heart, one for the GI system, right? So it'd be nice to have something that works for all. Well, how do we take that to the next step? So we found all these genes, right? How do you take it to the next step? So the next step is each gene encodes a protein, like collagen. So this is a picture of if you slice now the lung tissues and you check how much of that protein there is. So this is a protein called tenascin. And we looked at it in normal donor lungs, and we didn't see much of it. Brown is where it's present. The color of it is brown. When we looked at Scleroderma lung, so this is normal, you don't see much of it. When you look at a Scleroderma lung, you can see how much brown there is. So in Scleroderma lungs, there's too much of this protein, significantly more than what you would see in a normal lung. Okay, so what does that mean? So our next question was, well, if there's too much of it in the lung, is it spilling over into the blood? And can we see if there's too much of it in the blood? Because maybe we can use that as a blood test to sort of tell who has lung fibrosis, right? If they have too much of it in the lung because of lung fibrosis, and if it kind of leaks out into the blood, then maybe we can come up with an easy test without needing a lung biopsy. So that was the goal of that. And when we looked at that, we looked at the blood of healthy individuals. We looked at the blood of patients who had no pulmonary fibrosis, and then we looked at the blood of patients who had pulmonary fibrosis. You can see the levels go up in those groups. So we know now that patients with lung fibrosis have higher levels of this protein in their blood. So it makes it easy for us to get blood and check, are the levels higher? That means this person is at maybe higher risk of ending up with lung fibrosis. And to test that, what we needed is blood from patients before they had their lung fibrosis, and then blood after they had their lung fibrosis, right? And that tells you, was it low before and did it go up later? And that's what we did. We had a handful of patients where we had blood before they had their lung fibrosis, and then we had blood years afterwards when they had their lung fibrosis. And you can tell the levels went up between the two different visits. So that tells us that what we discovered through all these genes in the lungs may help us find markers we can test for in the blood that may tell us who's at higher risk of getting lung fibrosis. And then maybe those patients would get monitored more closely with more frequent pulmonary function tests or more frequent CT scans, right? So that's how you make use of it. And when we get treatments, it tells us which patients are at higher risk and should be on the treatment for lung fibrosis earlier to prevent it altogether. So that's the reason to do that kind of research. So here's another protein we found to be increased in the lung. It's called IGF-2. It's insulin-like growth factor. I don't expect you to know the names. I will not quiz you at the end today, just today. But basically, we looked at it, and again, we found that in the lungs, when you go from normal to patients without lung fibrosis to patients with lung fibrosis, the levels go up. So more of it in patients with lung fibrosis. So what we did is we took an antibody that blocks its effects of this protein. And this, if you just look at this dark band, that's collagen. The more of it you have, the darker and thicker the band, and the less of it you have, the thinner the band. So when we add this antibody that blocks the effects of IGF-2 to cells from the lungs, what we see is the collagen goes down. So this is elevated in scleroderma lung tissues, but when we try and block it, then the collagen goes down, suggesting that that might be a potential therapy for lung fibrosis. And actually, this antibody that blocks collagen has been in development by companies for other diseases. So that makes the case for, can we use it now for scleroderma? Can we try it in scleroderma? All right. We also looked at the role of genes and the environment in scleroderma. I'm sure you've heard of case reports. He being in New York, after 9/11, there are many cases of firefighters who were there who have developed scleroderma symptoms. So we know there's a link to the environment, but it doesn't seem like everybody who has scleroderma has been exposed to the same environmental factor, right? There's too many different things, like silica, like others. So we know that there are these occupational exposures, especially in men, to silica dust, trichloroethylene, for example, in the dry cleaning business, but a bunch of things that we know have been linked to cases of scleroderma. So what are we doing about it? Another research project we're doing now to look at the environment versus genes in causing scleroderma is what's called geographical clustering. We're looking for, through using what we call Geographic Information System, or GIS. You might have seen it in the Scleroderma Foundation e-letter. We were asking for people to fill out a survey to tell us where they live and where they lived when their symptoms developed. And so what we do is we take the cases and we use the software called GIS to put where they were when they developed their disease, and then find, are there regions where there's clusters of scleroderma patients? And then there are resources out there through the EPA where you can go and see, okay, well, what was in the air in that year in that region? And can we find the environmental trigger? So that's another way where, rather than working on the treatment, we're trying to work on what could have been the trigger or the cause. And thus the importance of participating in those studies, right? We can't get those answers if we don't get participation from the patients. All right, but when you look at environmental factors, we know that not everybody who gets exposed to them gets scleroderma. So we know that in Spain, many years ago, there was what was called the toxic oil syndrome. It was when 20,000 people ingested this adulterated oil that was bad, and there were a lot of cases of scleroderma-like illness. What was noticed, though, is it looked like the women there were more cases who were women than men of it, in different phases of it. So our question at the time was, if more women are affected, just like in scleroderma, there's more women than men, then is it hormonal, right? We have estrogen. That's sort of a hormone that we think is unique to women. Is that the problem? So we looked at that. And the way we looked at it is we took a human skin and we gave it estrogen. And when you give human skin estrogen, this is what happens to dermal thickness. The skin gets thicker. And if you block the estrogen with an estrogen inhibitor, you reduce the thickness. So we realized estrogen can cause fibrosis or skin thickening. We then looked, well, if estrogen can cause skin thickening, then do scleroderma patients have higher levels of estrogen then? Is that why they have the fibrosis and others don't? So we first looked at women. This is post-menopausal women who are not on hormone replacement therapy. And what we found that in patients with the diffuse skin involvement, there is higher levels of estrogen in their blood, right? But the gentlemen in the audience who have scleroderma will say, well, what about us? So we looked at the men, and what we found is men with scleroderma have even higher levels of estrogen than the women. And we know estrogen can trigger fibrosis. So now what we're doing is hoping to start a clinical trial using one of those inhibitors of estrogen that's been used in the setting of cancer to see if it would be beneficial in scleroderma and would reduce the fibrosis. But this is an example of how one piece of the puzzle led to another. And now here we are looking at a drug that's already FDA approved for cancer that we could potentially try in scleroderma. And being FDA approved is important because you can get to the clinical trial faster. The path is a more straightforward path than starting with something from scratch. Okay, so that's an example of repurposing of a drug that was approved for another indication, another disease that we could try in scleroderma. But we also know that hormonal influences, there may be other places where estrogen is high. People may not get scleroderma. There may be other people who ingested the bad oil in Spain and they didn't get scleroderma. So why is it that some people get it, some don't? So in occupational exposure, we know that people who are exposed to vinyl chloride in their work don't all develop scleroderma-like illness. Only 5% of them do. So why don't the other 95% develop it, even though they're exposed to the same thing, right? So that told us, is there a genetic predisposition? Is it possible that those with scleroderma have a higher susceptibility to getting it, but they only get it when they get exposed to the vinyl chloride or the toxic oil? And one way to test that is to test it in different cells with different effectors. And another way to do it, which is sort of a golden tool to look at what's the environment, what are the genes contributing to developing any condition, is to look at twins. So to look at the balance of environment genes, we did a study of twins. And we looked at identical and fraternal twins, because this is what's known in the field of genetics. If you have identical twins and 100% of the twins in both twin pairs both have the illness, then that means it's an inherited disease that we call autosomal dominant inheritance pattern. And fraternal twins, half of them both will have it in a twin pair, and half of them only one will have it. If it's recessive, then 100% of the identical twins will get it, and only 25% of fraternal twins will get it. That's just the inheritance pattern. And if it's an illness that's multifactorial, where you probably have the susceptibility, like we suspect in scleroderma, but that's not enough to get scleroderma, what you need is environmental triggers as well, environmental factor exposure, then you would expect, roughly half of the identical twins to both have scleroderma, and a very small percentage, maybe 5%, of the fraternal twins to have it, because fraternal twins are more like regular siblings than identical twins are. We looked at other diseases, other autoimmune, some rheumatic diseases like Crohn's disease, diabetes, MS, rheumatoid arthritis, lupus, and what it looked like is nowhere did we see 100% of identical twins, both twin pairs in a pair having scleroderma. And it was always fewer in fraternal twins, what we call concordant twins. So concordant means both individuals in a twin pair have it. Discordant means only one has it in a twin pair. So that told us that, in general, autoimmune rheumatic diseases likely are multifactorial. So you have a genetic susceptibility that makes you more prone, but then it takes other things happening in your life for it to start. When we looked at scleroderma, and we had identical twins, fraternal twins, we looked at patients with both the diffuse skin involvement, the limited skin involvement, and those with overlap. And we looked at identical and fraternal twins, and they were similar in age, similar in their disease duration. And then we looked at how many were concordant, both twin pairs had it, how many were discordant, and way more were discordant. So our concordance, 4 to 5% of our twins both have scleroderma. And in 95% of the twin pairs, only one has scleroderma. So what does that, who thinks it's genetic based on that information? Who thinks it's multifactorial with environmental influences? You're all paying attention. Not a single wrong answer, I'm impressed. Okay, so we now know that's what it is, right? When we look at autoantibodies in a positive ANA, it's a different story. Both have a positive ANA. So that told us that if you have scleroderma and a positive ANA, it is very likely that your twin will have just a positive ANA and not scleroderma. So why is your twin just having a positive ANA and you had the ANA and the scleroderma? And that's what we're working to solve. Okay, so the last piece of the puzzle that we're working on in the twins is what we call epigenetic changes, which are changes that happen on top of the DNA. We now know they were recently discovered. That's why they're interesting. We now know that there's many types of these changes, and I won't bore you with the names, but we know also what causes them. For example, in pregnant women, folic acid, the supplement, causes epigenetic changes on top of the DNA that are good ones for the baby. But a recent study showed that taking a lot of folic acid in men may be associated with autism in the baby. So folic acid is not necessarily good for everybody, for example. So it teaches you to differentiate. But many things, a lot of the environmental factors we just talked about that cause scleroderma can cause these changes on the DNA. They happen normally in the setting of aging, and they happen, like I said, folic acid, so diet can change them as well. All right, these are some of the environmental exposures that can cause those changes on top of the DNA. And basically, it's now such a popular field of study that it's being applied to all the diseases, including cancer. What's interesting about that is there are drugs that can change these epigenetic features that are on top of the DNA that have been approved for the treatment of some cancers. So again, the opportunity to take drugs that were developed for another illness and apply them to scleroderma. So what does the genetic study, the environmental study with the twins, and this epigenetic study taught us? It has taught us that patients probably have a genetic background that makes them more prone to scleroderma or other rheumatic diseases. And it's these events that happen through life, whether they be chemicals, whether they be viruses, we really don't know exactly what, but it's the things that happen through life, the exposures, and possibly the sequence, the order in which they happen, that determine, are you going to get autoimmune disease A or B? Are you going to get lupus? Are you going to get Sjogren's? Are you going to get scleroderma? But everybody probably starts with a genetic background that makes them more prone to it, and what they get exposed to in life decides which path they take. All right. So I'm going to end with why we continue to do the research we're doing, and what does that mean for the pipeline of treatments for the future. So a lot of the testing that we do in the lab involves these methods. What we call in vitro assays. In vitro assays is when we take a skin punch biopsy from you and we grow cells called fibroblasts in the lab, and we use them in experiments in the lab. So you take something outside the body and use it, like you take blood and purify the cells and use them in the lab. In vivo testing means you do the testing in animals, and the mice are the most common that we use in the lab, sometimes rats, but mice are certainly more common because they're the least costly. And then we do what's called ex vivo, which is testing things also outside the body, but keeping it as an organ, so not manipulating it in the lab. And the reason we go beyond that now is because most of the drugs that are tested and go to clinical trial are tested in mice. And I know for a fact that I can cure anything in mice. There's not a single disease you can trigger in mice that really cannot be cured. But probably 90 to 95% of the drugs that are effective in mice fail in human clinical trials. And this is not just scleroderma, this is in general, right? And they fail because we are not mice, and we are not going to respond to mice. So several years ago, I came up with a way to do additional testing that would make me feel more confident that a drug is likely to be effective in humans. So what I did is I took, and this was one of the very first experiments, I took human skin. So this is a human skin from a tummy tuck surgery. Aren't you glad I'm showing you all this after lunch rather than before lunch? So we took skin from tummy tuck, and this thin light-colored area here is the skin. That is the fat layer, okay? And we clean off all the fat, we cut it up, and we just keep the top layer of the skin, and we can put it in these dishes in the lab, plastic dishes, and the pink is their juice. So the pink is a liquid that has nutrients to keep cells and tissue happy and alive. And then we can inject it with things that we suspect cause fibrosis. And this is what you see. So we injected it here with a protein that we know causes fibrosis. And you can see as we inject more and more of this protein, the thickness of the skin gets bigger and bigger. That's fibrosis. This is a stain called Masson trichrome. This just picks up collagen. So the more you have of the blue color, the more collagen there is. So you can see where there's more and more collagen. But then we can inject it with things that we're developing as new drugs to show that now we can reduce or decrease that fibrosis. And the importance of now using this model, in addition to all the other models we use in the lab, is it helps us know that this is likely to work in a human because it worked in a human tissue in the lab, not just in cells, not just in mice, and it would reduce the risk of taking a drug to a clinical trial that has not been shown to be effective in a human tissue. All right, so a lot of things to test, but basically the take-home message here is all roads lead to collagen. Because what I showed you is there's increased collagen in fibrosis, right? That's one of the major proteins we look at. There's others, but that's the major one in fibrosis. And what we came across via a very accidental discovery in the lab is that another piece of collagen known as collagen 18, so the collagen we talk about in fibrosis is usually collagen 1 or collagen 3. There's more than 20 types of collagen in the body. What we found is a tiny piece from the tail end of collagen 18 can actually block fibrosis. So this is this piece of collagen. So remember, the darker the band, the more collagen there is. When we add this piece that we call E for endostatin, you see how the collagen is gone or shut down? So this is a piece from collagen that is helping us reduce or shut down collagen in the setting of fibrosis. So this is the newest molecule we're working with. Basically, what it does is it shuts down collagen and the other proteins. It stops the proteins from being linked. Remember, I told you the collagen in the skin and other tissues, they get linked, the collagen molecules, to each other. It stops them from being linked, which makes them more prone to being broken down and being taken away, and just reducing the fibrosis. So where are we at with this particular drug that we're developing? So we learned that it's unfortunately, a very expensive drug to make. To make it the traditional way, the synthetic way, it costs for every gram about $20,000 to $25,000, which is not reasonable, right? We don't want to bring another drug to patients that insurance is not going to cover and is going to cost them thousands of dollars out of pocket. But through partnering with people who know what they're doing with these things, what we're doing now is we are making it in plants. So instead of $25,000 for a gram, we can make it in plants for maybe $50 for a gram. And plants are unique, and I didn't know that until we got into it, in that you can express small peptides and proteins in plants very efficiently. The plant leaves, you can engineer them to make a lot of this peptide or protein for you. And what we do at the end is just chop the leaves. And no, you don't eat them in a salad, but we chop and we extract or purify the protein from them, and then it gets made into the standard sort of drug format. I actually had a chance to visit the facility where it's being made. The plant is a very distant relative of the tobacco family. So it's not going to be in salads, and it's not going to be smoked. Sorry to disappoint you, but it's going to be purified from it. And this is how the plants are grown. They're grown in these trays with a pink grow light, where the temperature from the bottom of these trays to the top does not vary from more than 2 degrees. So very controlled conditions to optimize for the growth of the plants. And our next step with this is to go to what's called a phase 1 clinical trial, where we look at the safety. Initially, you just look at safety by giving it to humans and making sure it's not going to be toxic. And often the way you do that is you do it in healthy volunteers. You give them the drug and you increase the dose of what you give them, and you just monitor to make sure there's nothing toxic happening, there's no toxicity. But we actually went to the FDA and had a great meeting with the FDA, and the FDA suggested to get there faster to do the phase 1 safety trial in patients with scleroderma. So we are now gearing up to start the phase 1 safety trial in scleroderma. And just like I showed you that this drug that we're developing in my lab is sort of getting close to the clinical trial phase, there are a lot of labs across the world, in the US and elsewhere, developing drugs as well that are promising. So if anything, I think this is the best time to be a scleroderma patient, because there are so many different drugs in the pipeline that are going to go into clinical trial, and I cannot imagine that all of them are not going to work. So there is hope that we will be getting an effective treatment soon, okay? So keep an eye out, and as you know, all the clinical trials get listed on clinicaltrials.gov as a website, so you can keep track of what's out there, what are the criteria to enroll in them, what sites have them. It's a good way to learn where they are. So this is sort of where we are with our research. This just gives you a sampler of how we're trying to address the pieces of the puzzle, what we're trying to look at, from environmental factors to genetics to repurposing of drugs to developing new drugs. So we're working at multiple levels. And that's why the research is so important, because you start with an observation in the lab and you end up with a drug and a therapy. I just hope that I left you at the end with the main message that finding a cure for scleroderma is not a mission impossible. Certainly not anymore. You know, maybe if you asked me 15, 20 years ago, I would have said, we're really far, we're not anywhere near there. But there's so much happening now in so many labs, so many things that are starting to come about that are on the horizon, that I think there is the promise of a cure. All right, this is my team. This is what it's like in Charleston. Right here is the building where the lab is. This is a tree we planted in memory of a young member of the lab who passed away last year, not from scleroderma, something completely different, but we usually take pictures by the tree so that the whole team we feel like we're all together again. And I will leave you with this last quote that I love from Mary Woodard Lasker, that I welcome you to use when your question is why it's important to support research and why is it important to fund research in academic institutions. If you think research is expensive, try disease. Thank you all for your attention.